Polyglossacarite completed. Do you hear me, anyone, please? Is it visible? This is one nucleotide. These are two nucleotides here. These are two nucleotides and they are linked. You see here how these are third carbon. First carbon. In this way, a molecule of water is released out so that extra linkage is formed. This is one extra linkage. The called phosphor.
স্যার নিচের পাটটা দেখা যাচ্ছে না।
হ্যাঁ কি ও ঠিক আছে। Now you see বলো
স্ট্রাকচার গুলো আগে কি আর কিছু
বল, কিচ্ছু শুনছি না আমি। বলো হরিত্র, হরিত্র বলো। In this way, molecular water is released out. Now again,
স্যার এটা একটু আবার এক্সপ্লেন করে দাও না।
কোনটা? Okay. Now you see the adenine is the nucleoside of adenine, then the nucleus. So together this one is the nucleotide of adenine. Already I discussed last day how nucleotide of adenine is formed. Now just below adjacent you will get another nucleotide, the same strand. This is one strand. Just below you will get another nucleotide. This guanine is the nucleotide of guanine. So these two nucleotides are linked by a phosphate. Actually, this one ester linkage, this is the other ester linkage, together called phosphodiester linkage. So you know that here two nucleotides are in line. Here the OH group present the third carbon of the sugar of one nucleotide, and the phosphate group presenting the 5th carbon of the sugar in the other nucleotide, involved in the development of an ester linkage to form a dinucleotide. As two ester linkages are found adjacent, the term used is phosphodiester linkage. Same thing happens in the other strand. This is the one strand of DNA, this is the other strand of DNA. And backbone is, this is the phosphate sugar. So you know the backbone of DNA is made of phosphate and sugar. Clear? দেখ বুঝতে পেরেছিস কিনা? Phosphate sugar. In this way, the backbone of the DNA is formed. You know, backbone of DNA is formed of phosphate and sugar by alternate arrangement of alternate location of the phosphate and sugar. So we just write phosphate and sugar. The two strands, the nucleotides, adenine binds to the thymine of the other strand and guanine binds to cytosine of the other strand. You know, adenine always binds to thymine by 2 hydrogen bonds. And guanine binds to cytosine by 3 hydrogen bonds. Therefore, the dot থাকতে বলা হয়. This is how a dinucleotide is formed. You can call this end is 5 prime, 3 prime, 3 prime, 5 prime. At one end of DNA chain, if the sugar is found to be with a free phosphate group, then it should be 5 prime, and in the other end of the same strand, opposite end of the same, you will get the sugar with the free hydroxyl group at 3rd carbon. So it is the 3 prime. So other strand, here you will get the 3 prime and here 5 prime. It means that two strands of DNA are actually anti-parallel. Two strands of DNA are found anti-parallel. Here, is it clear?
Yes, sir.
Now we get the structure of DNA. মুছে দিয়ে যা।
হ্যাঁ স্যার একটু দাঁড়াও।
আচ্ছা আচ্ছা ঠিক আছে। মনে রাখবে একটা বেসিক থিং মনে রাখবে, টু নিউক্লিওটাইডস adenonuclear linked by phosphodiester linkage, and you need to remember these bonds. So this is also a ester linkage. external linkage. Clear? হয়েছে বা সবার?
স্যার মুচলান না। স্যার উপরও জাগটা দেওয়া জানা তো।
বুঝতে পেরেছি কিনা? For angstrom. Actually, each step of ascent, each step this is the basic thing. At each step of ascent, the stand turn 36 degrees. So there are 10 steps. That's why it is 34 angstrom. The total turn. So the total 10 pages means number of ascent 36 into 10 is 360 degree turn. And here, as they are equidistant, so distance between two adjacent base should be 3.4 angstrom. This is the diameter, which is 10 to 20 angstrom. This is the structure of DNA. This is, you can call it 5 prime, 3 prime, 3 prime, 5. So this is the structure of DNA, double helical structure, and actually, this is ascent. Each ascent is represented by a pair of bases, and each step of ascent, the stand turn 36 degree. So, and 10 base, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 base involved. The total turn should be 36 into 10, 360 degree. And the length of which is 34 angstrom, and the diameter of the DNA is 20 angstrom. These are the double helical structure of DNA. 5 prime to 3 prime, one strand runs from 5 prime to 3 prime direction, whether the other strand runs from 3 prime to 5 prime direction. This is two strands of DNA are found anti-parallel. Now, in the railing, if it is compared to the ladder, in the railing you will get the sugar phosphate. They have to be linked again.
স্যার তুমি 36 ডিগ্রি লিখেছো কেন?
Huh?
স্যার লেফট সাইডে 36 ডিগ্রি লিখেছো।
Left side actually in each step, the ascent, the degree of ascent from 1 to this how much turn it cost 36 degree. From here to here, it is 36 degree. Are you getting me?
Yes, sir.
That's why you can call it. The total 10 bases, the total number of actually the turn should be 36 into 10, 360 degree. And length of which is 34 angstrom. It will be connected. বড় করে দেখাই।
স্যার আমিও পাগল হয়ে যাবো এটা দেখে।
হ্যাঁ কি হয়েছে? Look, this turning and then again they are leaving. বুঝতে পারছিস তো?
স্যার এটা কি G11C?
Triple bond. G always binds to C. A binds to thymine, adenine binds to thymine by 2 hydrogen bonds. Thymine binds to adenine by 2 hydrogen bonds. Guanine binds to cytosine by 3 hydrogen bonds. And cytosine binds to guanine by 3 hydrogen bonds. This is the backbone actually. This part is the backbone of sugar, phosphate, sugar, phosphate. So you can call it the backbone of the DNA, formed of sugar and phosphate. Is it clear? They're clear here.
স্যার একটু সাইড।
Sure.
স্যার স্যার তুমি জানি ডিএনএ স্ট্যান্ডার্ড 360 ডিগ্রি ব্যাপারে কি বলেছো?
স্যার একটু নিচে নামো।
Two strands of DNA are interparallel and is bound by the sugar phosphate sugar chain. By the sugar phosphate sugar chain. And nitrogen bases are found perpendicular to this backbone. Remember this thing. Nitrogen bases are found perpendicular to this backbone, more or less. কি বলছিলি?
স্যার 360 ডিগ্রি ব্যাপারে।
আচ্ছা, look. What is clear that it is found that in the chain of DNA represented by actually from this to this there is a angle of ascent. Clear? And it is measured that this ascent of each step is 36 degree. Okay, so if you calculate and they are equidistant, so then calculate again you will get another one. Suppose here from here to here, again the angle is 36 degree. So if you compare 10 base pairs, so what will be the measurement? If each step of ascent 30 degree, so 10 step ascent should be 360 degree. Are you getting me? 10 step of ascent, that's 10 turns. At each step of ascent, the stand turns 36 degree. So at 10 steps of ascent, the stand turns 360 degree. And the length of which is actually 34 angstrom. That means the each 36 degree length of place would be 34 divided by 10, that is 3.4 angstrom. So in this way actually DNA is helically arranged. You know, is it clear now?
স্যার শুধু টেনে নেওয়া হয়েছে কেন? অন্য নাম্বার নিতে পারতাম।
হ্যাঁ কি কি?
স্যার শুধু টেনে নেওয়া হয়েছে কেন?
না, actually দেওয়া আছে up to 36 degree. Are you getting me? In case of BDNA, it is taken up to 360 degree, sorry. It is calculated and 360 is at 10 degree. বুঝতে পেরেছো?
হ্যাঁ স্যার। স্যার নাইট্রেট বেসের ব্যাপারে কি বলছে? নাইট্রেট বেস,
হ্যাঁ
নাইট্রেট কি জানি বলছিলেন?
হ্যাঁ, backbone of DNA is made of sugar. Okay now. Now you see this backbone, that is sugar and phosphorus are found perpendicular to the nitrogenous base. That means if you go through them, they are found almost perpendicular to the nitrogenous bases and they are facing inside. বুঝতে পেরেছি দেখ।
স্যার বুঝিনি, আরেকবার বলো।
কোনটা কোনটা বল।
এখন যে বললে
আচ্ছা, look one thing. Phosphate and sugar, if you go through them, then they are actually the natural bases are here A, T, G, C. So you see their location. If you go to their structure, normally they are found perpendicular to the backbone of the DNA. Found perpendicular, এখানে বোঝা যাচ্ছে না এটা, found perpendicular, আমি ডাক্তার এখন দেখাচ্ছি, found perpendicular to the backbone of DNA. That means the way they are arranged, found perpendicular. Perpendicular means like this. So here I projected this side, you will get the S, so it is perpendicular to this. একটু ঘুরবে এটা। So found perpendicular to this. In this way, you will get almost perpendicular to the base, and they are facing down inwards. That's why they written the natural bases are found perpendicular to the backbone of the sugar. দেখো বুঝতে পেরেছি কিনা এবার। If you compare this backbone, they are found perpendicular to this backbone. দেখো, the backbone টা যে এখানে perpendicular হলো না, nitrogen projected almost perpendicular to the backbone হলো না, এটা দেখে তো বোঝা যাচ্ছে। দেখতে পাচ্ছ? Is it clear?
হ্যাঁ স্যার।
Okay. So this is what about the nucleic acid. এবার মুছে দেবো এটা। Yeah, I think it is clear to you what is 5' and 3'. At one end of the DNA chain, if the fifth carbon of the sugar is found to be with a free phosphate group, then it should be 5'. And the other, just opposite end of the same strand, you will get the third carbon of the sugar is with a free hydroxyl group. It is called 3'. Clear? Is it okay now? Mushiri? Mushiri Chire?
হ্যাঁ স্যার
I think I didn't discuss lipid. In this way, triglyceride is formed. Now, if one phosphate group is replaced, must not. Look, yes, you can call it simplest phospholipid. What happens here? One fatty acid is replaced by a phosphate group. Now it is clear? Now if choline is added to it, it should be look. This is called lecithin. This group is added. Actually, choline appears like this. Choline, the structure is and this weight an H-strong phosphate taking part in the formation of an ester linkage again. So this is the choline. এটা বোঝা গেছে? Now I am another diagram showing you. If
স্যার কোলিনের জায়গাটা কি করেছো?
look here. এরকম ছিল তো? Then you get it. বুঝতে পেরেছি এবার? দুটো মিনিট দেখো, just 2 minutes. I'm coming. Now if ethanolamine is added, how to show? Okay. তো হয়ে গেছে বোধহয়। phosphatidyl ethanolamine, and this phosphatidyl ethanolamine is also called caphalin. Clear? So this is what about the different fatty acids. দেখো এটা বুঝেছি কিনা?
হ্যাঁ স্যার।
This is all about the phospholipids. One is lecithin, other is the caphalin. Both are found in cell membrane also. And in this way it is formed. So you need to remember this thing. Again, ethanolamine means actually this is the ethanolamine. Clear? বুঝতে পেরেছো ভাই? Just look here how to form phospholipid. This is the triglyceride. This is actually the triglyceride. This is the simplest phospholipid where one phosphate group is replaced by a fatty one fatty acid is replaced by a phosphate group, then it is called phosphatidic acid. When choline is added to it, choline means then it becomes lecithin, and if ethanolamine is added to it, it becomes caphalin. In this way, both are phospholipid. Caphalin is also a clear? দেখো বুঝে দিচ্ছি। Now steroid nucleus. In short, it's a steroid nucleus. Is it clear? It is called The steroid is called cyclopentanoparhydrophenancing ring. ABC represent 3 financing rings. These are cyclopentan rings. That's why it is called cyclopentanoparhydro. This is here. Clear? So in all steroids or sterols or cholesterols or the sterols, the common feature is this: you should have this structure, the cyclopentanoparhydrophenancing ring, and then there should be an elevating chain added to it to form the different other cholesterol or cholesterol. In case of cholesterol, I am taking the same diagram here. One 8-carbon aliphatic chain is added to it. What is the 8-carbon aliphatic chain? Now you can call it cholesterol. Clear? When an 8-carbon aliphatic chain is added to steroid nucleus, the structure is called cholesterol. When an 8-carbon aliphatic chain is added to steroid nucleus, then it is called
So 8-carbon.
aliphatic chain. Is it aliphatic chain? 1, 2, 3, 4, 5, 6, 7, 8. Aliphatic chain is added to a steroid nucleus, then cholesterol is formed. Now this is the structure of cholesterol.
লাইনটা একবার বলো।
Whenever 8-carbon aliphatic chain is added to the steroid nucleus, cholesterol is formed. Lipids. So we discuss proteins, carbohydrates.
স্যার
হ্যাঁ বলো।
স্যার এলিফেটিক চেনটার মধ্যে 8টা কার্বন কোথায়?
এক, 2, 3, 4, 5, 6, 7 হয়েছে।
ও মানে সাইড চেন গুলোকে বদ্ধ হবে।
কিন্তু সিএসসি থাকে না, কিন্তু সিএসসি থাকবে। বুঝেছো?
হ্যাঁ স্যার এবার বুঝেছি।
Now if you are asked to write adenosine triphosphate, adenosine diphosphate, and adenosine monophosphate. That means it is ATP. It is ADP and it is AMP. Adenosine triphosphate, adenosine diphosphate, and adenosine monophosphate. That is ATP, ADP, and AMP. Can you draw these structures? Already you know the structure of AMP. While drawing the structure of AMP, you need to remember this. It is not deoxyribonucleic, so you have to draw the ribose sugar. मैं একটা দেখে দিয়েছি তাহলে বুঝতে পারবে কোলেস্টেরলটা মুছে দিই It is adenosine monophosphate. দেখ বুঝতে পেরেছিস কিনা? This structure is adenosine monophosphate. Clear? This structure is adenosine monophosphate. And when some other phosphate groups are added to it, yeah, for this you need to remember the ribose sugar is required to be drawn, not the deoxyribose sugar. I think you can remember this is the second carbon. So this is the adenosine monophosphate or AMP, and whenever you are going to add more phosphate group here in this line, for example, I am showing you. Look, so this is a triphosphate, and here you obviously will get the adenine base here. Look, now you can call it না ট্রাইফল নিতে হবে ক্লিয়ার Is it clear how triphosphate is formed? This is monophosphate is shown. And while doing triphosphate, you need to remember this. The adenosine triphosphate means here the sugar involved is ribose, not the deoxyribose. You need to remember this thing. কথা বুঝতে পেরেছিস?
Yes, sir.
Okay.
স্যার একটু সাইড।
লেখা আছে সবার?
হ্যাঁ স্যার।
না স্যার
আচ্ছা। Clear? Now, look one thing. If it is a diphosphate, now I have to draw diphosphate. You see? Diphosphate. Then what to do?
কমে যাবে।
This is diphosphate. Simple thing. But you need to understand this, how these structures are developed. Clear? Chapter, only one thing left. This is the enzyme part. I think everything is completed. The term enzyme was coined by Kuhn, K-U-H-N-E. Generally, enzymes are protein in nature. Exception are there are some ribozymes. Sumner said that enzymes are protein in nature. Exception ribozyme, non-protein enzyme.
স্যার কয়েন্ড বাই নামটা কি?
Kuhn, K-U-H-N-E. K-U-H-N-E, Kuhn.
স্যার সামনের সেট।
Sumner, other scientist, Sumner said that enzymes are protein in nature, but exception is ribozyme, non-protein enzyme. RNA with catalytic activity called ribozyme. Clear? Clear? Conception actually I was now enzymes basically whenever a protein is to act as an enzyme, it is to attain tertiary structure for this. Whenever a protein is to act as an enzyme, it is to attain tertiary structure. It is without attaining tertiary structure, no protein can act as an enzyme. Are you getting me? A protein, whenever it is to act as an enzyme, it is to attain tertiary structure. And without attaining tertiary structure, no protein can act as an enzyme. Clear up to this?
Yes, sir.
ওকে আজকে আমরা ইউটি ছেড়ে দিচ্ছি নেক্সট ক্লাস আমরা এখান থেকে শুরু করবো এখান থেকে ক্লাস কোথাও যাওয়া যাবে না আসলে মানে আমরা এখান থেকে এনজাইম ক্লাস থেকে শুরু হবে নেক্সট ক্লাস আমি বলেছি টু স্টার্ট ফ্রম দা এনজাইম ক্লাস
ঠিক আছে স্যার
Tomorrow I start with the enzyme class. Okay, thank you very much.